first one will be Dr. Kathleen Knight with the USDA Forest Service, and she'll be presenting on the impacts and management of emerald ash borer. So if you want to welcome Kathleen.
All right, well, thank you so much for having me here today. Is this volume okay? I feel like I'm really loud. All right. Thank you so much for having me here today. I'll be starting with an overview about ash, and then I'll talk about why it's so important and talk about my work over the last 20 years to understand impacts of emerald ash borer on our forests and study management strategies. So ash is ecologically, economically, and culturally important. These photos show a green ash wetland in northeast Ohio, a beautiful huge white ash growing in northwest Pennsylvania, and a blue ash in central Ohio. There's 16 different species of ash in North America. We have 5 of them in our part of the country. And they all have different habitat preferences, different ecological preferences, and different roles in their ecosystems. Of the 5 species that we have, the first is white ash. This is a species that typically lives in more upland forests mixed with hickories and maples and oak trees. The second is green ash. This is a species that's particularly important in floodplains and riparian areas, where as part of a healthy forest, it regulates hydrology and helps prevent flooding. Black ash, Fraxinus nigra, is a more northern species that does get down into parts of Ohio, and it is really important in wet ecosystems. So this is a picture of Cedar Bog State Nature Preserve here in Ohio in early spring when all the marsh marigolds are in bloom before the ash trees have leafed down. Pumpkin ash, Fraxinus profunda, or if you read older literature, Fraxinus tomatosa, is also a species that inhabits wetter areas, and it's more of a southern distribution, but it gets into Ohio as well. And then finally, blue ash, which is a really neat one. It likes high calcium soils, so it typically grows in places with limestone outcroppings or limestone rocks near the surface. It's such a cool species. It's got these weird 4-angled kind of winged stems, kind of like a euonymus almost. It's a really unique and beautiful species. So ash inhabits this key niche in northern floodplain forests, especially the green ash and the black ash, where this is a unique species that combines cold tolerance, it can live in those northern habitats, flood tolerance, as well as shade tolerance. And there are very few species as you get further north that have those characteristics. It helps regulate the hydrology of those wet forest systems. All of these ash species help sustain biodiversity. They support quite a few lepidopteran species. They're also culturally important to Native American tribes. Black ash basketry is especially culturally important. It's also economically important. Timber of white ash is used for tool handles and baseball bats, things like that. The emerald ash borer is native to China and was accidentally introduced to North America. The adult beetles will be emerging soon in the spring. They leave D-shaped exit holes in the trees as they emerge. They fly off, feed on leaves, mate, and lay their eggs on the bark of the ash trees. When the larvae emerge, the larvae burrow in just beneath the bark and start feeding on that layer beneath the bark that we call the phloem. And as they feed, they create little tunnels beneath the bark that we call feeding galleries. And the picture on the right is one of these EAB larvae. That's the stage that really does the damage and kills the trees. This is a picture of a tree with the bark removed, showing just how many of those feeding galleries, when you get a really large infestation, these emerald ash borer larvae can make. And so as they feed and create these galleries, it disrupts the flow of sugars in the tree, disrupts the flow of water and nutrients, and the tree quickly dies. Emerald ash borer was first introduced in the mid-1990s and discovered in 2002 in southeast Michigan. Since then, it's spread throughout the region. It was detected in northwest Ohio in 2003 and has spread throughout the state over the following 10 years. It's now been in Ohio for 2 decades and has killed billions of ash trees throughout our region. In order to manage forests that are impacted by emerald ash borer, we first have to understand the impacts. And those are going to be different depending on which species of ash you're talking about, which kind of ecosystem you're talking about. We also need to understand the long-term dynamics of ash and emerald ash borer. And then we can think about combining different strategies to achieve specific goals to kind of mitigate those impacts. So that might include breeding and restoring ash with resistance to emerald ash borer, conserving ash genetics, either through insecticide treatment or seed collection, releasing biocontrol insects, doing genetic conservation of ash, either through seed collection or insecticide treatment to preserve trees, and restoration of systems to maintain ecosystem function. So along with several collaborators listed here, I've been tracking the impacts of EAB in long-term monitoring sites in Ohio since 2005. These plots are in metro parks, state lands, and private lands, typically a small area of forest, either surrounded by agriculture or suburban areas. There were over 3,000 ash trees in the plots, representing all 5 species that are native to Ohio. We tracked them individually over time. We rate the health of each ash tree on a scale of 1 to 5, where 1 is healthy and 5 is dead, and 2, 3, and 4 are stages of decline. So, and then we take the average of those health ratings for each site. So the blue and dark green dots are healthy stands of ash trees, and as you get into the red dots, those are mostly dead stands. So this shows 4 different years, and you can see in 2008, dead ash showing up in northwest Ohio. By 2011, the mortality had spread quite a bit. By 2014, it had moved through central Ohio. And by 2019, we had dead ash in most of our sites, except for a few blue ash sites, which I'll talk about later. This QR code is a new publication that came out last year that has kind of a great summary of all of our ash monitoring information over the last 20 years in it. So I'll give people a second if they want to snap a picture of that, because I'm really excited about it. All right. Emerald ash borer is capable of infesting trees 2 centimeters, about an inch or larger, but it seems to prefer trees that are at least 3 or 4 inches within trunk diameter. So here's the diameter distribution of trees before and after mortality from EAB. When I'm talking about diameter, it's the trunk diameter at breast height. So think about, hugging a tree. That's the diameter that we're talking about. The left graph is 2007 before EAB, and this is the initial diameter of the trees in centimeters from 0 to 90 centimeters. That's almost a meter wide tree up there. And then in 2019, after emerald ash borer had come through. And what you see is the small size class from 0 to 5 centimeters, 1 or 2 inch trees still remains, but all of these other size classes have been greatly reduced. So it just wiped out all of those larger trees on the landscape. There are some species differences in what happened with these larger trees. There are very few surviving larger black ash. It got hit fairly hard. And almost all of the surviving ash that's larger than 30 centimeters is blue ash. This chart shows those ash trees larger than 4 inches trunk diameter, and in that right column is the survival, the percent of them that died by 2019 in our plots. So white ash, black ash, and green ash were hit really hard. They all have more than 90% mortality at that point. The blue ash, though, had much lower mortality in 2019, only 28% mortality, which is still terrible, but not quite as terrible. This is a graph just kind of showing what's going on with blue ash. This is the longest infested blue ash site of my sites in southwest Ohio. It had both blue ash and white ash growing in the forest. On the y-axis is that canopy health rating from 1 to 5. So 1 here at the bottom would be healthy and 5 is completely dead. So these are the mean for each of those species at the site. And the white ash here in orange, you can see in 2007, mostly, fairly healthy, about a 2. And then fairly quickly by 2009 or 2010, all of those trees were dead. The blue ash, on the other hand, stays fairly healthy for a few years, but then after the white ash is dead, we start to see the blue ash declining over time. And of my 6 sites that had blue ash, 4 have graphs that look like this, where the white ash dies first, and then the blue ash slowly starts to decline, and we do see mortality, we do see exit holes, we do see, all of the typical symptoms of EAB. The other 2 of the 4 sites, the blue ash persists and has stayed healthy. So I think the jury is still kind of out on what that long-term trajectory is for blue ash. But this really makes the case for the importance of long-term data. Because if you just looked at a snapshot in 2009 or 2010, you would say, white ash is susceptible and blue ash is resistant. Blue ash isn't going to have any problem with emerald ash borer. So it's important to have that longer-term perspective. In addition to tracking the mortality of the large trees, we also tracked seedlings in the monitoring plots. In many of our Ohio sites, there are tons of ash seedlings persisting in the understory. These were too small for that first wave of EAB, and in places where gaps opened up, some of them grew really rapidly. That's not the case everywhere. We see that a lot in Ohio. In northwest Pennsylvania and the Allegheny National Forest, there's almost no regeneration. So when those large ash trees died, white ash was just gone from most of the forest. The seed bank is very short-lived. Only about 2 to 3 years after massed seed year do we see new seedlings coming out of the seed bank. So what we're really left with here in Ohio is kind of this orphaned cohort of small ash trees in many of these sites. We also track emerald ash borer populations over time using purple panel sticky traps at several sites in northwest Ohio and central Ohio. So this is a picture of a bunch of emerald ash borers stuck to our sticky trap. Here's the trap hanging in the tree. You may have seen these at some point for detection or monitoring. And here's the results from those sticky traps. This shows the population dynamics of emerald ash borer over time. So on the y-axis is the number of EAB we caught on the trap. Northwest Ohio, which was infested first, is the green circles, and central Ohio is the blue triangles. You can see that EAB populations peak during that first wave. They peak around the time that we hit 50% mortality of the ash trees. And then the EAB populations crash as all of the ash trees die because they have eaten all of their food source and they have nothing else to eat. Unfortunately, they don't go away. They stick around at fairly low densities, maybe persisting on those smaller trees as they get up to susceptible size or on the few remaining ash trees in the sites. And then more recently, we've seen kind of another small second wave in many of our sites where we did have surviving trees or where smaller trees had grown into larger size classes. In terms of what this looks like long-term for ash populations, there are some surviving ash out there. We have a small percent of large trees that survive at some sites. We've identified quite a few healthy green and white ash in that 4 to 10 inch diameter range, as well as some much larger healthy blue ash. Those healthy large trees form the basis of our resistance breeding program. They might have resistance that's genetically heritable that allows them to survive emerald ash borers. So we're really interested in those trees, and I'll talk more about that work in a few minutes. There are also a lot of smaller ash, of course, that we've talked about that are too small for EAB. Those are still remaining in those sites and growing long term and getting up into those susceptible size classes. And we are seeing mortality of both those larger trees as well as the smaller cohorts that are growing during the second wave of EAB that we're experiencing now. Oh, I wanted to tell the story. So this is my team. This is the first surviving large ash we found in Northwest Ohio. We had just spent the day out in the forest recording data on hundreds and hundreds of dead ash trees, which was really depressing. And then on the way back to the car, as we're walking back, our summer intern Kyle spotted this beautiful, healthy, large ash tree right by the road. Mm-hmm. And so that was the first one we found, and it gave us a lot of hope. We were really excited. We went back and talked to Jennifer Cook at the lab, the geneticist who's leading the breeding programs, and we continued, that really launched us doing complete surveys of some of these sites to really look hard for these surviving trees. So management implications of this work that we've done on EAB impacts. We have seen that ash mortality and EAB population dynamics follow a very predictable pattern. This allows managers to plan different actions: underplanting, removal of hazard trees, removal or treatment of invasives. So that's helpful. Other states have learned from what we experienced here in Ohio and are more prepared for what's coming. Emerald ash borer does remain a threat. It has not gone away. And although most of the ash trees over 4 inches DBH do die, there are some large, rare trees that have survived. So how do we respond to invasive pests like EAB? It's likely to have devastating ecological and economic impacts. So how do we mitigate those impacts? The answer is to combine multiple strategies to achieve reduced pest impacts. And this is basically a schematic for what we call integrated pest management. I'm going to briefly go through kind of each of these areas and how it applies to emerald ash borer, what we're doing in those areas. And then I'll give a quick overview of our work on resistance breeding and restoration. So we can do preventative measures, slowing or preventing the spread of emerald ash borer, encouraging people to burn it where they buy it and not remove firewood, doing risk assessments. Cultural control, which includes resistance breeding. This is a picture of a healthy, surviving large ash tree right next to a dead one at one of our sites. Biological control. There are many introduced parasitoid insects that actually parasitize emerald ash borer. Some of those are native, some of those have been brought over from Asia and released to try to control emerald ash borer. Chemical controls with insecticide treatment to protect trees. Monitoring, of course, is an important component of any of these strategies. And then mitigating that impact of emerald ash borer either through restoration plantings. This is a Dutch elm disease tolerant American elm planted next to some dying ash trees in a wetland forest, or through ex situ and in situ genetic conservation, so seed collection or treatment of trees to preserve ash on the landscape. And so the hope is that by combining these different strategies in situation-specific ways, we can respond to EAB, reduce those ecological and economic impacts of this pest. So I'll now give a quick overview of our tree resistance breeding work. So the general process starts with these rare, large survivor trees in forests where lots of trees are dead. We collect material from those large trees, we collect branches and twigs, propagate those trees to make genetically identical copies, and then we test those to identify which ones have resistance. Once we identify the ones that have resistance, we can either go ahead and plant them in locally adapted seed orchards or breed them together to make even more resistant trees to put in the seed orchards. Then those seed orchard trees breed with each other, and the seeds produced can produce trees for restoration or reforestation. The way we're doing this with ash is Jennifer Cook, I should say, leads this program. I collaborate with her on this work. We find ash that are in areas that have been infested by EAB a long time. They're large enough that they were a susceptible size during peak EAB. They have a healthy canopy at least 2 years after that mortality rate leveled off. So this is that healthy tree there again. We do top grafting with the twigs. So these are the twigs from the survivor trees grafted onto rootstock in pots. Either that or we do bud grafting onto the rootstock. That allows us to make a bunch of genetically identical copies of the trees in pots. We also plant some of them out in archive plots, and we also do controlled pollinations. That's our team up in lifts, bagging branches to keep other pollen out so that we can do controlled crosses of those trees to see which ones make good parents and produce good progeny that have resistance. And then the way she actually tests them for resistance, there's 2 different ways. One is the EAB egg bioassay, and the other is using field trials. With the bioassay, those potted trees, she actually puts emerald ash borer eggs that are on filter paper produced by our colleague Therese Poland onto the trees. And then when the egg hatches, the ash borer burrows into the tree. She lets it go for about 8 weeks and then peels the bark of the tree to see what happened to the larva. With a susceptible tree, we end up with a big, fat, healthy larva, right? But with a tree that has resistance, the tree actually kills the emerald ash borer larva, which is awesome and what we want to see. Not all of our lingering ash we're testing have resistance. About half of them seem to be able to do this. The best ones have killed about 45% of the larvae. To compare, the susceptible ones kill 5 to 10 or 12%. Our top 10 lingering ash average only about 19% kill. So it's enough to allow the tree to live longer, but they're still at risk of death. The other method, the field trials, she plants them out and just lets them get naturally infested, and that's to have a comparison of its field performance versus that egg assay. So what happens if we take 2 of these best lingering ash that seem to do a really good job of killing larvae, they have moderate levels of resistance, and we breed them together? So this is a graph. Jennifer's done that about 27 times, produced 27 families of progeny from different crosses. And each bar here is a different tree from one of these crosses. On the y-axis is the percent of larvae that were killed by the tree, from 0 to all of the larvae being killed by that tree. And you can see there's a lot of variation, right? There's some that did not do a very good job. They did not inherit the good genes, right, from their parents. They were pretty susceptible. But there's others that did a lot better than their parents, and that's what we want to see. So the exciting thing here is that this green arrow represents the best parent. And so we have a lot of trees better than the best parent. And this blue arrow represents Manchurian ash, which is the Asian ash species that coexists very happily with emerald ash borer and can survive with emerald ash borer in China. So we are getting trees that are at that level of resistance, which is awesome. So the idea is to take these best trees then and to put them, so this represents all of these crosses and progeny, and we take the best progeny and put those in the seed orchard. And then they'll cross and create progeny that have those high levels of resistance. And Jennifer's already started 2 different seed orchards. One that kind of encompasses this region in the orange circle, one that encompasses the region in the red circle. The first is just lingering ash clones, those surviving ash clones, and the second is those best progeny from the crosses being planted together. So we're encouraging people to watch for large survivor ash trees as well as elm trees. We also do an elm breeding program that's very similar, and submit them to our database. You can use this app called TreeSnap to submit them to us, and we prioritize over time the trees each year that we're going to propagate and test. So we're working through those submissions. I'll say a couple quick words about our genetic conservation work. So the idea of genetic conservation is to preserve the genetic diversity of ash before it's killed by EAB. So we can do ex situ genetic conservation, which is just seed collection and storing those seeds in climate-controlled facilities so that they last a long time, or in situ genetic conservation, which is basically protecting trees on the landscape using insecticides. We initiated a study on the Allegheny National Forest in northwest Pennsylvania to do insecticide treatment of trees and to monitor the impacts of ash borer there. So this is kind of a different landscape than Ohio. Instead of smaller parcels of ash, this is a very large forested landscape. Unfortunately, even in that large forested landscape, we saw something very similar to the map of Ohio, right? So in this instance, emerald ash borer started on the south end of the forest and kind of worked its way north. And you can see with that ash canopy condition, those red dots kind of spreading across the forest as emerald ash borer makes its way through. So by 2021, most of the ash on the forest was dead. In 2015, the ANF received funding to treat 500 ash trees with insecticide to conserve genetic diversity. So they contacted us and wanted to know what the best way was to arrange that treatment to conserve the most genetic diversity. We formed an advisory group to design that treatment strategy, and we chose 27 large plots across the forest where ash was still healthy, and randomly selected 20 trees in each plot to treat every 3 years with the imidazole benzoate trunk injections. In those plots, there were somewhere between 22 and 215 ash trees. So if we treated only 20 of them in each plot, that left somewhere between 2 and almost 200 untreated ash trees. And so you can see here, they were particularly close together, so it made for a nice picture with a treated and untreated tree, but often they're much further apart as well. And one of the cool things that we're looking at is what happens with those untreated trees. Do they receive some protection just from being close to a treated tree, just from being in those treated tree plots? And that's what we call associational protection. Several studies have demonstrated protection of those untreated trees in a variety of different contexts, including Autumn O'Brien, who did some work here in Five Rivers Metro Parks with the tree treatment program that was here. And so this is showing that we did have confirmed that we're seeing some degree of associational protection. So on the y-axis is that 1 through 5 canopy condition, with 5 being dead, 1 being healthy. The red dots are untreated trees in untreated plots across the forest, so no insecticide at all. And what we see with those is, of course, nearly complete mortality of those trees over time. The green triangles are the treated trees that got the insecticide in the treated plots. And so the insecticide was able to successfully kind of stabilize the health of those trees and keep them healthy over time. The yellow circles are the untreated trees that are in those treated plots near the treated trees. And you can see that they're doing a lot better than the untreated trees elsewhere in the forest. Many of them have stabilized. We did have some of them decline and die. We're continuing to monitor this over time to see if this continues to work out long term. But that really increases your bang for your buck if you can only treat some of the trees and protect a lot of other trees by doing so. What we found from the insecticide treatment was that we did some genetic studies to show that we actually conserved 97% of the genetic diversity on the forest by treating only a little over 500 trees across the forest. So we were really thrilled with that. We figured out how to maximize efficiency of your treatments, to reduce costs and maximize genetic diversity that you're conserving as well. In conclusion, understanding those impacts on forest ecosystems and long-term population dynamics of emerald ash borer allows for management planning. We do have those rare surviving ash trees that have resistance to EAB, and I'm really excited about that work. And there are a number of other management strategies out there to help reduce the impact of this really destructive pest. And I wanted to quickly thank funding sources and the many partners who have helped us with this work, and the many sites that have allowed us to set up these long-term monitoring plots on their lands, including Five Rivers Metro Parks. We've also had many teams of summer interns. I think at this point it's over 60 summer interns have contributed to this work. And with that, I can take a few minutes to take any questions. Yeah.
Two questions. One, has any genetic studies been done to try to identify the source of the resistance?
Yes. So they have done genetic studies to try to figure out genes. They're also looking at trying to figure out rather than genetics, they're doing proteomics to look for individual proteins and stuff. So there are a number of people who are collaborating with Jennifer who basically use the material from her program to study these differences. They've found a few different proteins that are potentially involved at this point. But yeah, so they are starting to look at that.
And my second question is, in terms of the protection offered by treated trees and untreated trees, any idea of the mechanism of that? Is it insecticide investing into the roots?
We don't know. So we're trying to figure that out. We thought maybe it would work like herd immunity with vaccination, and that the more trees you vaccinate, there would be some kind of threshold amount. So when we set up that study, there were somewhere between 2 and 200, untreated trees. And we thought we'd see somewhere along that it would break down and the untreated trees wouldn't get protection. And it didn't. Treating 15% of the trees works just as well as treating 90% of them in a plot. So that was really surprising. So it doesn't seem to be operating like vaccination. Otherwise, you could vaccinate 2 people on your block for COVID and everyone would be protected. So it's not working that way. We got a sub-meter accurate GPS last summer and GPSed each tree. So we're going to look at that question of spatial proximity to kind of get at whether it might be spreading to the roots. So if that's the case, we would expect to see just the trees that are really close to an untreated tree get that protection, and trees that are far not get it. So we're analyzing that right now, and hopefully we'll have a better answer on that. But that's one of the things we're looking at. The other possibility is that we have inadvertently created lethal trapped trees, which were used in the slow ash mortality programs. So trees that were already kind of declining when we started treating them and were attractive to emerald ash borer and drawing them in, and then we treated them to make them lethal, may have drawn in a lot of EAB and killed large numbers of EAB, kind of protecting the whole plot. So we're kind of looking at that as well to see if our data would support that as a potential mechanism. But yeah, it's been a surprise. It doesn't work the way we thought it might. So we're looking into it. Yeah.
I almost hate to ask, but the non-native parasites that you brought in, how do you test those to make sure they're not going to kill a good bug?
Yes, that's definitely concerns of how do you test those non-native parasitoids. So APHIS did a lot of testing of those insects on an array of native insects, an array of economically important insects before releasing them. And they're tiny, tiny parasitoid wasps, so they're not like big wasps that sting people. So they're not a threat to humans. They did find that I think one of them occasionally parasitized a native ash borer. But our native ash borers are kind of in trouble anyway if we lose all of the ash. So the others didn't parasitize any native species. We didn't see any other parasitism of native species. So they really do seem to be very specific to emerald ash borer from that testing. You can't test everything, so you kind of cross your fingers, but it's not it's not a situation like the cane toads where we throw it out there without any thought process ahead of time. So they're trying to do their due diligence to look into that before they put them out there. Yeah.
You said the seeds only remain viable in the seed bank for about 3 years. So how long does the controlled environment extend the vitality?
So far, they think they can keep them viable for at least 20 years and maybe more. So when they keep them in these perfectly, humidity controlled and cold, freezers, they can keep them a long time, and they pull some out every once in a while to test viability, and they're still viable, the ones that they started collecting back at the beginning of the collection program. Any other questions? All right, well, I will turn it over to Donna.